Hello everyone and welcome to Instant Biology by Dr. Neelam. Today we are going to continue the series on immunology, and this is the next lecture of the series, Basics of Immunology. In the previous lecture we had already discussed about precipitation reactions, and in today's lecture we would be concentrating upon agglutination reactions. So basically what we are doing is we are talking about antigen antibody reactions. This is the second one, and in this we would be talking about agglutination reactions. If you have not seen the precipitation reactions video, I would recommend you to browse through my playlist of immunology, and you can find the precipitation reaction video over there. So let us talk about agglutination reactions. What is the difference between precipitation reaction and agglutination reaction? First of all, you need to understand precipitation reaction. What was the precipitation reaction? In that reaction, what we were doing is we were mixing the antibodies with a soluble antigen in an aqueous environment. Okay, soluble antigen. The antigen was soluble. It was solubilized in the aqueous solution, and you were mixing antibody from outside. What happened is precipitation resulted because of the formation of lattice. Okay, there was a lattice structure formation between the antigen and antibody, and because of that later on the precipitation happened, or precipitate was formed. So this was seen. But in today's lecture, when we are talking about agglutination reaction, what happens is that over here antibody is interacting with particulate antigen. Okay, so now you can see the difference between precipitation and agglutination reactions. Over here the antigen is of particulate nature. What do you understand by particulate nature? Particulate simply means that like a particle. Okay, particulate means just like a particle. Okay, so particles means that this would not be soluble inside the aqueous environment, or it would not be soluble in whatever environment it is present. That means it is non-soluble. Particulate form, it means it is not soluble. Antibody plus particulate antigen is giving you visible clumping. Okay, so the concept is almost the same. The nature of the antigen is different. Over there precipitation reaction was occurring. Over here agglutination reaction is occurring. So you would ultimately see a visible clumping in agglutination reaction. So what would be the eventual result? It would be a visible clumping. Okay. Now let us talk about the antibodies that cause this kind of a reaction. If you remember my precipitation reaction video, you would recall that the antibodies that were used over there were precipitin antibodies. We gave them a particular name, and the name was precipitin antibodies. But in this case the antibodies would be called as agglutinins. Fair enough, because the reaction name itself is agglutination reaction, so the antibody name should be agglutinin. Fair enough, no problems in that. Next is cross-linking of polyvalent antigen happens. What is happening over here? That the antigen would be polyvalent. You would remember from the precipitation reaction video also, it is good that the antigen should be polyvalent, because what happens when it is a polyvalent antigen, what happens is that multiple antibodies cross-link to the same antigen, causing the precipitation or agglutination or clumping. Basically it will come down, right? So that is why the antigen should be polyvalent. Valency of the antigen should be high. So what I mean by this is that if I say that this is the antigen, what I mean to say over here is that suppose this is an antigen that I have drawn, and it is having multiple binding sites, multiple epitopes that could be recognized by different antibodies. When this is done, it causes a cross-linking between different antibodies and different antigens, and because of this what happens is visible clumping is seen. Concept remains the same, right? Over there also we were seeing a lattice and clumping and precipitation. Over here also we are talking about cross-linking, right? Now the next concept is that we have seen that excess of antibody causes inhibition of agglutination reaction. What does this mean? This is true for precipitation reactions also, that whenever you use excess of antibodies, that means high number or high amount of or high concentration of antibodies, you would not see agglutination reaction, or this would inhibit agglutination reactions. What did I say? This is true for, this concept is true for precipitation reactions also, as well as agglutination reactions also. This is true for both of them. Okay, and this phenomena is called as Prozone effect. It is called as Prozone effect. Now, what is Prozone effect and why does this occur? This is important. You should be knowing, you should be aware about what is Prozone effect. Remember, I'm talking everything that I'm talking about is from QB. Okay, let us talk about Prozone effect. What is Prozone effect? If it is asked what is Prozone effect, then you can clearly say that Prozone effect is the effect that causes the inhibition of the agglutination or precipitation reactions because of the increased amount of antibodies, right? Now, what happens or why this Prozone effect takes place? Whenever antibody concentration is high, so I've marked an arrow in the upwards direction. What does that mean? The concentration of antibody is high. Now you see, if the concentration of antibody is high, the number of sites of antibody that would want to bind to an antigen would be high. So antibody binding sites would be high. By antibody binding sites, I clearly mean that the sites that are present on the antibody that wish to connect with the antigen, they would be high. Higher than what? Higher than the number of available epitopes. What are epitopes? I've already discussed this in one of the first class or second class, I don't remember. So epitopes are the regions on the antigen to which the antibodies bind or recognize, right? So if the antibody binding site is greater than the available epitopes, why? Because the antibody concentration you have kept very high. What would be the result? So the result would be univalent binding. Univalent binding. What does that mean? Suppose this is an antigen, it would be binded by this antibody only. So why do we care? It has binded, right? Why do we care? It has binded. This antibody has bounded to this antigen. But I'm saying that there would be univalent binding, no cross-linking would be seen. So why cross-linking is important? You see, now let us say that because some other antibody also would have binded to this, and this antibody might have also been connected to this antigen, just like this. Just like this. I mean, I'm not drawing the complete diagram, but you can see that to this antigen only there are two antibodies that are binded. There is a cross-linking that you can see. If this cross-linking is not present, there would be no visible clumping only. This would not come down, this would not clump. Okay, so what we are saying is that whenever the antibody concentration would be high, univalent binding or no cross-linking can be seen. There would be no cross-linking. If there would be no cross-linking, ultimately what would be the result? There would be inhibition of the agglutination reaction. There would be no agglutination reactions. Okay, so how can you diagnose this? You can diagnose it by varying antibody or antigen concentration. What you can do is you can make multiple dilutions. Suppose this is the highest concentration of the antibody, this is a diluted version, this is again diluted version, and so on and so forth. You are again and again and again and again diluting. Okay, let me just draw it and show it to you. So what I'm saying is, suppose this is one test tube, diluted, diluted again, diluted again, diluted again, and you have mixed some amount of antigen over here, a particular amount of antigen, same amount of antigen to all of them. Now since you are diluting the antibodies, what will happen at a given concentration? At a given concentration, now suppose this is 1, this is the complete antibody, highest concentration, this is 1/2, 1/4, 1/8, 1/16. What will happen? You are continuously diluting, isn't it? you are diluting the antibody, and at a given concentration the antibody would be so low in concentration, that it will start binding to the antigen. And the inhibition that was present because of the Prozone effect, it will be removed. The inhibition would be removed because the concentration of the antibody would be so much decreased. That is why I have written that can be diagnosed by varying antibody and antigen concentration. I believe this would be clear. Now, this Prozone effect can also occur because of polyclonal antibody. Polyclonal means, monoclonal means binding to the same antigen. Okay, monoclonal. But if I say polyclonal means there are different types of antibodies that are binding to different epitopes. So in case of polyclonal antibody, there can be IgG also, there can be IgM also, there can be all sorts of antibodies. Okay, so what happens when you are using polyclonal antibody, what happens is the IgG, immunoglobulin G, what happens is this immunoglobulin G that goes and binds to the antigen, but it cannot cause agglutination reaction. It goes and binds to the antigen, but it cannot cause the agglutination reactions. Why? Because there can be two reasons. I would be coming to that. But these types of antibodies that are binding to the antigens but are not resulting into agglutination reactions, this kind of antibodies are called as incomplete antibodies, right? They are called as incomplete antibodies. And because of these antibodies, because of these IgGs, what happens is the binding site of the antigen is occupied, right? Suppose this is the antigen. Now the IgG has come and binded over here. Now this binding site is occupied. This binding site is occupied by this IgG, this IgG. There are better antibodies over here that are called as IgM. There are better antibodies, but now they cannot come and bind over here because this site is already occupied, right? These IgM can definitely cause agglutination reactions, but they are not able to cause agglutination reactions because they are not able to bind over here because the site is already occupied, right? So the antibodies that cannot cause agglutination are called as incomplete antibodies. They result in blocking of the IgM. Next, what is the reason that these IgG, these antibodies are not able to result in agglutination reactions? The reason behind that can be because they have restricted flexibility in the hinge region. So if you remember, right, so this complete region is called as the hinge region. Okay, if I draw this antibody, this region is called as hinge region. Now this hinge region is responsible for creating lesser or greater angles between the antibodies, isn't it? Now you can see this is the hinge region is responsible for creating greater or lesser angles. Now this greater or lesser angles is required for proper binding with the antigen. You can relate with that. Now there is a restricted flexibility. There is these hinge regions in case of IgG. Maybe the reason behind that is the hinge region because of the IgG, they are not very flexible. They have restricted movement. Because of that, they are not able to properly bind to the antigen or the epitope of the antigen. Next reason is that the antigen itself has the binding site in a very closed pocket, or let us say that the density or distribution of the epitopes are in deep pockets. Now let us say that this is the antigen, this is some kind of a protein, and this is some kind of a protein, or this case some kind of an antigen. Now let us say that the binding pocket is over here. Now you can see that this is a closed area. Binding pocket over here is interior or closed area. So there are chances that the antibodies might not be able to reach over here, or they might not be able to bind to this region. That is why it is called as, that is why it might result in the inhibition of the agglutination reaction. I hope this is clear. Now what are the examples of agglutination reactions? So we have talked about what is agglutination reaction, what is Prozone effect, the reason of Prozone effect. Now let us talk about examples of agglutination reactions. Blood typing, this is one of the most common examples of agglutination reactions. Whenever you go for a blood testing and the individual or the technician, he or she tells you that you have a blood group of such and such type, then what is he or she doing? He or she is doing the agglutination reactions, because you know that RBCs, suppose this is the RBC, this is RBC, and on the RBC there are certain antigens that are present. Now these antigens determine what type of blood group do you have. Okay, suppose you are having this A antigen, then you would have the blood group A, or this is the antigen A, blood group A, antigen B, blood group B, antigen A B, blood group A B. If there is no antigen, then the blood group is O. This you already know. I am not interested in talking about all of this. I just wanted to give you an example of blood typing, and this is quite simple, so I'm not going into the details of that. But now let us come to the next point. Bacterial agglutination is used for diagnosing or diagnose infections. Now bacterial agglutination is also one of the techniques, and what you do in this bacterial agglutination is that you take the serum of an infected individual. Now suppose if an individual is infected by a bacteria, in bacterial infection, now if the individual is affected by the bacterial infection, his antibody or his serum would comprise of antibodies that would have been made because of that particular antigen only, isn't it? The antibodies would have formed because of that particular antigen only. And whenever you are using those antibodies, you have obtained those antibodies from the infected patient, and you want to test whether the infection that this person is having is of that bacteria, that particular bacteria only, what you can do is you can directly allow the interaction of the antibodies present in this infected individual with the bacteria that you have suspected. If they are interacting well, if they are forming the agglutination reaction, then you can predict that yes, the individual was suffering from that bacterial infection only. That means it can help you to diagnose a bacterial disease. There are a number of bacteria and the symptoms are almost the same. Suppose you are having headache, suppose you are having fever, suppose you are having sneezing, etc., etc. The symptoms are almost the same. How would you come to know that whether the individual is suffering from such and such kind of bacterial infection, or this kind of bacterial infection, or that kind of bacterial infection? This can be one of the important tools through which you can identify, because antibody is very specific for a given bacteria or bacterial antigen. So whenever they would combine, then only the agglutination reaction would be positive. Okay, so this is clear. So that means that bacterial infection can be identified. This is what I have written over here. You can easily identify bacterial infection, plus you can also identify the different species. Suppose Salmonella typhi, there are different species of Salmonella, okay, Salmonella typhi being one of them. So you can easily identify that also, that species also. One more important concept that I would like to share over here is antibody titer. What does that mean? What is titer? Now suppose that I want to make dilutions. I am making dilution. Suppose I obtained the serum or the antibodies from the patient. Okay, I have obtained the sera or the antibodies from the patient. Again, what I am trying to do is, this was the first. Again I have diluted. Again I have diluted. Again I have diluted. Again I have diluted. This again I am diluting again and again and again and again. And what I have done is I have put particular amount of bacteria inside each of the tubes. Particular amount of bacteria or bacterial antigen, whatever might be the case. So the highest concentration of the dilution, okay, the highest dilution that you have made, suppose I am making the dilution one by, in your cube A, one such example is made. Let us say that this was let us say 1 by 1, this was 1, this was 1 by 2, this was 1 by 4, this was 1 by 8, this was 1 by 16. So what you are doing, you are increasing the dilution. In over here you are increasing the dilution. So the highest dilution that still gives the agglutination clumping, agglutination reaction, that is taken, and you make the reciprocal of it, or you make the inverse of it. That is called as the antibody titer. Let us take another example. Let us say that at 1 by 640, let us say that at 1 by 640 you can see the agglutination reaction. This is the dilution at which you can see the agglutination reaction, and when you increase the dilution to the next level, that is 1 by, if you multiply 640 into 2, you would get 1 by 1280. That means this is the another twofold dilution. Okay, next twofold dilution this is, but you are not able to see the agglutination reaction over here. You are able to see the agglutination reaction over here. That means you would not consider this, you would consider this. And what would be the antibody titer? It would be 640. Okay, so using this antibody titer, you can also predict that what kind of what kind of bacterial infection is present inside the body of the patient using this antibody titer. I believe you would have a clear concept of antibody titer now. You would have clear concept about what is bacterial agglutination and how is it used to choose or find out what is the infection. Now another concept, or the last concept, let us say, is the agglutination inhibition reaction. So what is the example of this agglutination inhibition reaction? The basic example and a very important example is the early kits that females used for testing out whether the individual, whether the female is pregnant or not at home. So early types of home pregnancy test kits, they are using this agglutination inhibition reaction. Agglutination inhibition reaction. Now let us just rub this out and let me explain you what is agglutination inhibition reaction. So as the name suggests, what is or what might be the agglutination inhibition reaction? In this reaction you inhibit the agglutination to happen. You are not allowing agglutination to happen. Okay, you are not allowing the agglutination to happen. So what is that, or how does that occur? So there is a kit. I'm going to take the example of a kit and explain you with that only. So there is a kit that is used to predict the pregnancy, whether the female is pregnant or not. So what is the kit, or what all does the kit have? So it has a latex bead. Now this round thing, this thing is the latex bead. This is a bead like structure made up of latex, and on the bead these are HCG, human chorionic gonadotropin. These HCG molecules are present. Now you would be knowing that all the pregnant females, whenever a female is pregnant, the HCG is secreted. Okay, so already we have this HCG attached on the latex bead. Plus what we are attaching over here is anti-HCG antibody. Anti-HCG antibody is present. Inside the kit, anti-HCG antibody is present. Latex bead on which the HCG has binded, that is present. What is the test procedure? Now whenever a female is pregnant and she wants to test whether she is pregnant or not, she collects urine, her urine, and she tests it with this kit. So anti-HCG antibody, and this is incubated, and HCG carrier conjugate is checked out to observe whether the visible clumping is present or not. So leave this, I am going to show you that how this is checked. Okay, this is just the test procedure. I am going to show you that how this agglutination inhibition helps us to find whether a female is pregnant or not. So whenever there is a negative reaction, that means female is not pregnant. Whenever there is a negative reaction, you see that the female is not pregnant, what will happen? So you can see this is the latex bead. Okay, so I'm not drawing the entire structure over here, I'm just showing you, I'm just writing things. So this was the latex bead, and on this latex bead there were HCG. There were HCG, and what you have done, there is anti-HCG also. There is anti-HCG also. Anti-HCG antibody. Anti-HCG antibody is present, and this is the latex bead plus HCG that is present. Latex bead LB plus HCG. Now when the female is not pregnant, if the female is not pregnant, there would be no HCG in the urine. No HCG in urine. If there is no HCG in the female's urine, what will happen? This latex bead and this anti-HCG antibody they will combine and they would form agglutination reaction. Or you can say visible clumping. So you would see a visible clumping if the female is not pregnant. Why? Because this latex bead plus HCG and anti-HCG antibody, they combine together, they agglutinated, and you would see a visible clumping in case that the female is not pregnant. In this case where the female is not pregnant. So this is the case. Now if I say that the female is pregnant, positive reaction, female is pregnant, positive reaction, and female is pregnant. So what will happen? I'll draw again. So LB, latex bead, plus this is all HCG that is attached, LB plus HCG, and there is anti-HCG antibody also, anti-HCG antibody is also present, and apart from that there is human chorionic gonadotropin that is also present. HCG from the female urine is also present. Why? Because the female is pregnant. If the female is pregnant, there would be HCG molecules from the urine also. So this is HCG from urine. Now what will happen? This HCG from the urine and anti-HCG antibody, they would combine together just like this. They would combine just like this, and this latex bead plus HCG would not be or would not show visible clumping because they are combining by themselves. This latex bead and HCG, they are not binded by this anti-HCG. Instead what happens is this anti-HCG antibody is now binding to the HCG from the urine. This is free, so there would be no visible clumping. No visible clumping. So if there is no visible clumping, agglutination will not happen, and you have, what you have done is you have inhibited the agglutination. Agglutination inhibition is seen in this kind of a test. So this was all about agglutination, about the applications, about the reactions. I believe you would have liked this video. If you have liked, just press the like button, hit the subscribe button, and also don't forget to press the bell icon so that you can get all the notifications whenever we are posting a new video. Plus I would also like you to join my Telegram channel so that you can get all the latest and recent updates. Thank you so much and have a good day.